Hello, my dear students. I hope you all have been doing well. From today, we are going to start our new topic, and it is about lipid metabolism. In lipid metabolism, we will be having three lectures, lecture number 7, 8, and 9. And last week, we have completed carbohydrate metabolism. In that, we almost cover six lecture that was based on the carbohydrate metabolism, means its absorption, digestion, and then the metabolism. So here in this week we are going to cover the metabolism of lipid. And in this means we have to discuss here in this week, we have to discuss about the lipids. And I will deliver three lectures on for this topic, and lecture number 7, 8, and 9. Lecture number 7 would complete in whole one lecture, and lecture number 8 would be based on three parts. Part 1, lecture number 8, part 1, part 2, lecture number 8 part 2, and lecture number 8 part 3, and then we would be having lecture number 9. So in lecture number 7 we would be discussing about the digestion and absorption of the lipids. This is the introduction, and then after that we would be in lecture number 8 we would be discussing about the metabolism of lipid. In that first part we will be having a small means introduction of both catabolism and anabolism of lipid, and second part we will be discussing about the catabolism of lipid and then in the third part we would be having, I think so, um, beta oxidation, and then we would be studying about the anabolism of lipid, that is the synthesis. So this was the whole means outline of the this week lecture, that is lecture number 7, 8, and 9. So from we are going to start from here. We already have been study about the carbohydrate, digestion, and absorption. So like as the carbohydrate were the macromolecule, here the lipids are also larger macromolecule, and lipid are made up of the fatty acid monomer. As in in our previous topic we studied that carbohydrate polysaccharide are the carbohydrate that are made up of monomeric, means glucose monomeric unit. So here lipids are the larger macromolecule, and that these macromolecule are made up of fatty acid monomers. And the lipids are generally not water soluble, they are insoluble in water, so their digestion and absorption is somewhat little bit difficult as compared to the other biological macromolecule. And here we first before starting from absorption and distribution, first we have to a little bit have an idea that what are the lipid, and what type of lipid metabolism we are means what type of a lipid we are going to discuss here. So lipid are macromolecule, they are made up of monomeric fatty acid monomer. So what does it mean? So here I would define you that lipids are having three types. They may be triglyceride, they may be phospholipid, or they may be present in the form of esterols. So here we are taking the example of cholesterol, and that is one of the type of esterol. And here we are having an example of the phospholipid, that is the membrane lipid, it found in the membrane. And the here we are having an example of glycerol, that is the one of the triglyceride. Here we are having an example of the triglyceride, that is one of the type of a lipid. So first we have to understand that if we understand their basic structure, so we easily understand their absorption, distribution, and metabolism that are coming in the my coming lecture. So here we can see that what does mean by lipid are macromolecule and they are made up of monomeric fatty acid unit. So here we can see this is the macromolecule, lipid macromolecule, and it is made up of monomeric fatty acid. So here we can see if we are talking about the uh, triglyceride, that is one of the lipid, one of the macromolecule, macromolecule. So we can see here it is having a head that is made of glycerol backbone or glycerol moiety, and this glycerol moiety attaches with three fatty acid monomer or three fatty acid units. So these are the monomers that connect with each other via the glycerol backbone and make up a big macromolecule, that is the triglyceride. So the concept is clear here. So in case of phospholipid, by definition, that phospholipid is one type of the lipid and they are made up of uh, excuse me, they are made up of fatty acid monomeric units. So here we can see this phospholipid is having a head, and this head is having a phosphate group, and that is attached to a glycerol moiety, and then this glycerol moiety is attached to two fatty acid monomeric unit. So all together, this is the lipid macromolecule, that is a phospholipid. So due to this phosphate group, this head of this phospholipid is behave as hydrophilic in nature, and these two fatty acid chain are hydrophobic in nature, means they cannot soluble in water. So here we can also see that this phospholipid is attaching with two fatty acid chain, and one is saturated without any double bond, and one is unsaturated with having double bond. And if we talk about the cholesterol, so cholesterol is an example of the sterols, and this is the means you can say this is the formula, this is the structural formula of this cholesterol. So and the cholesterol, the basic means importance of cholesterol or esterol is that the all form of esterol contain a cyclopentano perhydrophenanthrene ring that is attached with the hydro and this uh, ring in different type of esterol attaches with different functional group, but in the form of, if this ring attaches with a hydroxyl group, so this we called is as cholesterol. So cholesterol is a esterol, that is a type of fatty acid, that is a type of lipid or fat, and it is having a cyclopentano per hydrophenanthrene ring and having a hydroxyl group as a functional group. So this is whole about the means what are lipids. So actually this is your previous course that you have studied in 311, course number 311, but before starting I wish to recall you so you can easily understand the upcoming discussion. So what happened in the absorption and digestion and the metabolism, digestion, absorption and metabolism of fat, as the carbohydrate, protein and other macromolecule, the macromolecule cannot absorb or cannot, when we digest means we eat or we take the fat in our body from outside, so they we take in the form of macromolecule. So if we will be taking the fat in our body, so we will be taking in this form, either it will be triglyceride, either it will be phospholipid, or it will be cholesterol. Okay, whatever food we are taking from the outside, so the fat that is present in that food would be either in the form of triglyceride, either in the form of phospholipid, either in the form of cholesterol. So these are all macromolecules, so they need digestion. They need digestion to absorb them into the cell. And after absorption of these macromolecule in the cell, they need metabolism to produce energy. So what is the digestion, what is the absorption, and what is the metabolism? So when we are taking any food, and these means triglyceride, phospholipid, and cholesterol are present in that food, so we what would be now happen first? They digest. So what is the digestion? Digestion means we will be having some enzyme or some in our body. So what they in different compartment or different organ of our body, these enzyme will break the bond between these monomeric units and convert this polymer into monomer. Okay, so for example, if we are having some enzyme for the different, we are having different enzyme for the lipid digestion or triglyceride, phospholipid, and cholesterol digestion in our body. We will be discussing in next few slides. But here you have to understand why we would need digestion, absorption, and metabolism, and how it produce energy. So what happen if we are taking this lipid in our body, in our food, in the triglyceride form or in phospholipid form or in cholesterol form? So what happened first? What first, there would be digestion that would be takes place in the different parts of our body, the different organ of our body, that break these bond among these bond in the macromolecule that are present between the monomeric unit of this macromolecule. So what happened? This triglyceride would convert into diglyceride or monoglyceride eventually. For example, if an lipid hydrolyzing enzyme cutting breaking the bond from here, so what we will be having, we will be having two unit, one will be diglyceride, one will be monoglyceride. So and then if we will again break this bond by the action of a enzyme, any lipid hydrolyzing enzyme or fat hydrolyzing enzyme. So what we will be having? We will be having three glyceride. So after the conversion of this triglyceride into the monoglyceride, this monoglyceride would then absorb in our cells. And then after the absorption, what happened? They reaches inside our cells, and in these cells further means the metabolism takes place. And what happened in the metabolism? In metabolism, different functional group would add up in this. For example, monomeric triglyceride, monomeric monoglyceride, that will be the monomer of this polymer or macromolecule, or different functional group will remove, and then means this monosaccharide would modify, and eventually means its bonds will break. And what we will be having, we will be having a modified molecule that would produce energy in the end. So that energy we would be using for to perform our work. Or this is about the catabolism. But in metabolism, the reverse thing will takes place, and we would be synthesize larger macromolecule from the monomeric unit. So the same thing would takes place with this type of lipid, that is phospholipid. That when we will take this phospholipid in our body, so what happened? That the different enzyme would means act on this phospholipid, will break the bond, and then the convert after the breaking of bond, this phospholipid would convert, means this fatty acid chain would remove, means separate from each other, and then we will be having means monomeric form of this phospholipid. So the same would happen with this cholesterol. So here in this lecture, we are going to discuss about these absorption and distribution. First of all, digestion and the absorption uh, uh, of these type of lipid. And in the end of this lecture, we also uh, we would discuss that how these lipid would transport in different part of our body. So here we are taking an example of our body organs, and um, this slide is just a need to use to explain you how in what part of the body these type of lipids we are talking. Just keep in your mind that we are talking about the digestion and absorption. We are right now talking about the first digestion of the lipids, and then digestion of the lipids. uh, in this lecture we are first talking about the digestion and absorption of the lipids, that is triglyceride, phospholipid, and cholesterol. So we are talking about these three type of the lipids. So where the digestion takes place, main digestion means in three part of our body, or three organ of the body, digestion of lipids takes place. It starts from the mouth, and some of the lipid digestion takes place in the stomach, although very little it would be. And the rest of the lipid would be digested in the intestine, small intestine, and the absorption would takes place in the intestine. So these are the organ where the digestion and absorption takes place. So actually in my previous slide I told you that the lipid are fat soluble molecule. They are not water soluble molecules. So what is the and the lipids are digested with some difficulty as compared to the carbohydrate. So here the question is this: if the lipids are fat soluble, and the all enzyme, actually the all enzyme that is present in our body, these are water soluble. So how it could be means it could be possible for the water soluble enzyme to work on the fat soluble fat soluble uh, lipids? Because they are having two different medium. Lipid are having fat soluble nature, that are fatty fats in nature, fatty acid in nature. However, enzyme are water soluble in nature. So for any molecule or any enzyme or any macromolecule to work on another type of macromolecule, so they should be means having same medium. If one molecule is a water soluble, so another other should be water soluble. So because of their same nature, they would mix with each other gently, and then would work on each other gently. But here is the problem with the lipid, that lipid are the fat soluble, and water is the and enzyme are the water soluble. So how question is this: how it could be possible that the fat soluble, the lipids that are fat soluble, digested by enzymes that are water soluble? So this is the means main question of this lecture, and in our next slide we are addressing this question. We are understanding how it could be possible. So first of all we would be seeing that in which compartment of the body or which organ of the body what type of enzyme present that helps for the digestion of the lipid. So this slide is here I make a conclusion of, you can say, the whole lecture, whole topic of the digestion of the lipid. So this slide is showing here that we are having three compartments in our body for the lipid digestion, that are triglyceride, phospholipid, and the cholesterol. And these three compartments are the mouth and the stomach and the small intestine. And in the mouth, what type of the lipids are digested? These are the triglyceride and phospholipid. And what chemicals or enzymes are present here, and what other physical or chemical help present here, that enable a lipid to digest. So here in this compartment, that is mouth or our buccal cavity, here we are having saliva. Here we are having the digestive enzyme that is lingual lipase that help to digest the lipids. And here actually what happened, the what the phospholipid that we take in our mouth from outside as lipid during our food or during our meal. So these phospholipid actually act as emulsifier. So in our coming slide we would discuss that how these emulsifier help for the um, digestion of the lipid. So we have to just keep in mind that in mouth we will be having emulsifier like phospholipid, we are having the enzyme lipase, we are having the saliva that help in the digestion of lipid. And uh, we and what other thing help for the digestion of food in the mouth? Chewing action of our teeth. So it also help in the digestion. Actually, whenever we talk about the digestion of any macromolecule, either protein, lipid, fat, so two type of digestion takes place. And what are these two type? One is the chemical digestion, and other and second is the one is the chemical digestion and other is the you can say physical digestion. So the chemical digestion is would takes place with the help of the chemicals, that is the enzyme and maybe other chemical. For example, if we talk about the carbohydrate metabolism, we are also having the different juices in our stomach that help in the carbohydrate digestion. So we are having two type of digestion for each means macromolecule. One is the chemical, other is the physical. So chemical is the physical or mechanical. One is the chemical, other is physical or mechanical. So a chemical is the with the help of the enzyme or with the help of the chemical. And the physical, actually we should have to adopt the particular means more precise term for the digestion for the physical digestion, that is chemical digestion. So two type of digestion takes place, one is the chemical, other is the mechanical. So chemical is with the help of the chemicals, that may be the enzyme or other type of chemical that is present already in our body or we take from outside with our food. And the mechanical digestion is that is our, for example, our contraction of our means stomach muscles, or mechanical digestion is the means chewing action of our teeth. So these are the mechanical digestion or the peristalsis movement of our intestine. So in our mouth, what is the mechanical digestion? Mechanical digestion is performed by the chewing activity of our teeth, and in the or the contraction of our mouth muscles, or in the stomach the churning movement. One type of movement that is takes place in our stomach is the churning movement. Churning movement of our stomach, that is the muscular movement of our stomach cell. And in the intestine, peristalsis movement of our intestine. So these are the type of the mechanical digestion that help for the digestion of each macromolecule, including the lipids. So we have discussed that in mouth what happened, what type of mechanical and chemical digestion takes place, and what chemical um, and mechanical sources help there. And the in stomach, how what type of mechanical and chemical digestion takes place, and what type of the mechanical and the chemical sources help here. So here is the stomach gastric lipases help for the chemical digestion, and the churning movement of stomach and stomach muscular contraction help for the mechanical digestion, and the emulsifier that help for the digestion are the diglyceride. So diglyceride help for the digestion, for the for further digestion in stomach. So what happened in the small intestine, we are having the peristalsis movement of the small intestine that act as a mechanical digestion, and then we are having here bile acid in a small intestine, that actually small intestine does not release this bile release from the pancreas and enter in this small intestine via pancreatic duct. So this bile having the different component like bile salt, uh, lecithin and the different means drive cholesterol, means different substances that drive from the cholesterol and phospholipid, or you can say these are different substances that are the derivative of the cholesterol and the phospholipid, and they act as the chemical digestion, and the pancreatic lipases are present in the small intestine that help for the chemical digestion. So this was whole the means conclusion of the mechanical and chemical digestion, and the sources that help for the mechanical and chemical digestion of lipid in different part of the our body organ. And so moving to the next slide. Yeah, so here it is a whole the picture, and in actually I am having different slides in coming, means in the rest lecture I am having the different slides, and these slides are actually the explanation of this picture. So here in this picture we are seeing that how the digestion of the lipid, triglyceride and phospholipid, start from the mouth and end on the small intestine, and how the fat absorb in the small intestine and then transported into lymphatic vessels. Actually the whatever the digested fat that digested throughout this alimentary canal reaches to the small intestine, and after that from the small intestine it transported to the it means accumulate in the lymph node, and from there from the lymph vessel it transport to rest of our body. So here we are having means point 1 to point 6. So actually this is the means stages of digestion that takes place throughout our body. So one by one we will discuss about all these points. So first is the digestion of the lipid, that is triglyceride and the phospholipid and the cholesterol in our mouth. So we will see what happened in our mouth. In our mouth what happened, whatever the food that we take in our body from the outside. So what happened, it first come into contact with our saliva, and what happened, we chew this food by means of our uh, teeth, and this is the mechanical digestion. And after that what happened, the phospholipid that we take from outside in our mouth, means that are present in our food. So you have to keep in your mind that whatever food we are having from outside, it is having the fat, and fat is including both, it is having the triglyceride and it is also having the phospholipid. So what happened, that these fat including both type triglyceride and phospholipid would come into contact with your saliva, and your tooth will means break down this food with the help of saliva. Actually what does saliva do? Saliva help to make your food slimy, so it can be easily cut into small pieces. So what happened after that, triglyceride will be present like that, that is present in your saliva. But what happened, the phospholipid act as emulsifier. What is emulsifier? Emulsifier is an additive, means that an additional part of your food, you can say, that help to mix one component into the other component. So how this phospholipid is acting here in as emulsifier, that phospholipid is helping this to mix this triglyceride with the enzyme of you that is present in your mouth. So what are the enzyme, what type of enzyme present in your mouth for the digestion of the fat in your mouth? I already explained in previous slide, and here also written that these are the lingual lipase that are present in your mouth, and they help to digest your food lipids, triglyceride. So what happened when you will take your food? So how this triglyceride that are lipid soluble would mix with the enzyme that is lingual lipase. These triglyceride mix with the lingual lipase with the help of this emulsifier that is phospholipid. So why phospholipid is acting as emulsifier, why it is necessary? This is the answer of my question that we discussed in a very previous slide, that if the fat are fat solubles, not soluble in water, and the enzyme are water soluble, so enzyme has to work on triglyceride to make them digest, to make means to digest them. So if the two things uh, means have to work with together, but they are opposing each other, how it could be possible they work together? They cannot work. So we need to have a third party that allow them to mix with each other, and this third party is the phospholipid. They that in our means biochemistry term we say that emulsifier. So this emulsifier would already present in your food as phospholipid, and it help to mix triglyceride with that is the water fat soluble means component of your food to mix with the water soluble component uh, 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 water soluble component of means the you can say that water soluble component of your mouth that is already present in your mouth that help to digest this triglyceride. So when this triglyceride mix with this lingual lipase that is an enzyme by the help of this phospholipid, so this lingual lipases would easily start their work on these triglyceride. And then what it do, then this triglyceride would easily digest it. So this happen in your mouth, and after the action of this lingual lipase on triglyceride, what happened, you will be having teeny droplets of the fats that would be easily separate from the watery component. So what happened here, how digestion takes place in your mouth? In the first instance, when you are taking your food in your mouth that are having the fat, for example triglyceride and phospholipid, so what happened there? Here we can see here what happened there. In mouth you are having triglyceride, you are having phospholipid and in mouth you are having the digestive enzyme. So at first instance, phospholipid act as emulsifier, enable lingual lipase to mix with fat that is triglyceride. So what happened, this triglyceride that is the fat easily separate from the um, uh, easily separate as droplet from the watery component of your mouth. What was the watery component? That was the saliva that was present in your mouth. So this happened in your mouth. And what happened in the next after that, what happened, this whole content means the fat droplet would reaches into your stomach. Although in stomach um, very little digestion of fat takes place in stomach, but however we would study that what will happen then in stomach with your with the fat. So in the stomach, when this triglyceride would reach in the stomach. So what happened, the gastric juice would means the work on triglyceride and then break this triglyceride into diglyceride and the fatty acid. Okay, so in my previous slide, I have explained you what are the triglyceride. Triglyceride are the fatty acid molecule that are having three fatty acid chains and one of the glycerol head. So what happened, I explained you in my previous slide that one, if we will go move on to the previous slide. So what happened in the stomach, in the stomach we are having this triglyceride, it is having this three chain fatty acid chain. So what gastric lipases do there, gastric lipases just break this one bond, and what we get after the digestion, this digestion in the stomach, we get the diglyceride, means a whole, this means molecule that is having 240 fatty acid chain, this green and the pink one, with this half glycerol unit with 2 carbon. So we would be having this as diglyceride, and the one fatty acid chain will be removed, means separately. So we would say this, that after the action of gastric lipases on triglyceride, we would be having the as a result or as a product, we would be having the diglyceride, uh, these two fatty acid chain with this three, I'm sorry, I told you previously that 2 carbon, not 2 with all 3 carbon. So after the action of the gastric lipases on this triglyceride that is present on in your stomach, we would be having as a result this diglyceride that is having the 2 chain with this glycerol head, and only one chain would be separate, uh, 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 lonely, and it will be called as fatty acid. So it would happen in your stomach. And what happened next, that this is the chemical digestion that will takes place in your stomach. And what happened in your how and what happened next, uh, what happened, this diglyceride um, would then enter into small intestine. So you are getting an idea what happened in the mouth, and then just we would be having a separate droplet molecule by the action of lingual lipases, and these droplet actually that are containing pure, you can say, lipids, reaches to the your stomach, and in stomach the gastric lipases does their work, and then what happened, this triglyceride in the stomach convert into diglyceride, and this diglyceride then enter into your small intestine. So what happened in your small intestine, in small intestine you are having means you are having different juices that come from your liver. We call these juices as the bile or bile that are containing bile salts, lecithin, and different derivative of cholesterol. And so these are actually synthesized in your liver, stored in pancreas, that we also store in pancreas, and then uh, store in gallbladder, and then move into a small intestine by pancreatic duct. So this was these juices what how help in the digestion. These bile, what do they do again, this bile act as emulsifier. Okay, so how the bile act as in emulsifier, these bile act as an emulsifier and help um, intestinal, what enzyme we will be having in intestine. So in intestine we are having pancreatic lipases. So what happened, these bile that would reach from the liver into the small intestine, it is having the um, it is having the bile salts and other components. Actually these bile salts would help emulsifier and how it acts as emulsifier in intestine, we will be having diglyceride. So you keep into the mind that diglyceride is not means completely digested, means your lipid are not completely digested until it convert from triglyceride unit to the monoglyceride unit. So the monoglyceride would be the end product of the digestion. After that it will absorb. So what happened in the mouth, you are having triglyceride, then in intestine you are having, in stomach you are having triglyceride, that convert into diglyceride. In mouth you are having triglyceride, in stomach you are having diglyceride from the triglyceride. So this diglyceride reaches to your intestine, and in intestine this diglyceride would further have to digest. So what happened, how the diglyceride digest, because diglyceride is also water insoluble, uh, and it is a fat, and it has to be digested. So how it could be digested, because the enzyme that are present for its digestion in the small intestine are pancreatic lipases, or as I said you previously that enzymes are water soluble, so they can only work on the water soluble molecule. So what happened, how these the enzyme pancreatic lipases can work um, on the diglyceride if these two things not mix with each other. So how we this problem would solve, this problem would solve by the bile acid that would come from your liver in the form of bile, and it is containing different bile salts, lecithin, different derivative of cholesterol and phospholipid. So all these means a component of bile um, uh, act as a emulsifier, as a additive, help to come in contact both part, means a water soluble enzyme with the water non soluble fat, that is the triglyceride in the present in the intestine. So after that what happened, so we have so after that what happened, that after the what happened when means with the help of these bile salt, these two things means diglyceride come in contact with the pancreatic lipases, and what then pancreatic lipases would work on the diglyceride, and they separate diglyceride from the glycerol head. So what we will be having? We will be having two separate fatty acid chains, because diglyceride was would be having two fatty acid chain with a glycerol head. So as we can see here, diglyceride would be having two fatty acid chain with a glycerol head. So what happened in the intestine we are having gastric lipases, we are having bile acids or bile salts. So what happened, these bile salt uh, make help these diglyceride to contact with the gastric lipases. When gastric lipases come in contact with the diglyceride, so these gastric lipases break this bond and remove this glycerol head from these two fatty acid chain. So in the intestine we would be having these fatty acid chain after the digestion. So it would be, we will go again, so it will happen in a small intestine. So so actually what so so actually what we will go. Yes, so actually what happened in the small intestine, the pancreatic lipases convert diaglyceride into monoglyceride. So this would happen in our small intestine. And what happens So this was the one work of the bile, that the bile help in emulsification of diglyceride with the pancreatic lipases, and after that the pancreatic lipases will work on the diglyceride and would convert the diglyceride into um, monoglyceride, or remove the glycerol backbone from the fatty acid chain. So this was the first one work of bile. Bile will perform another work. What is this other work? Bile would help this separated monosaccharide to easily transport from the intestinal microvilli cell to the cell where they have to absorb. So how the bile perform this work? Bile would do uh, bile would envelop the fatty acid and monoglyceride and form a micelle. So what this micelle do? So what does this micella, what is this micella? Actually, this micella is the, uh, uh, it is a droplet. You can say this is a droplet of uh, monoglyceride. So here we can see that we are having a um, this we are having a mm, so So what does this micelle actually is? Actually, these are bile salt help to together means help fatty acid chain monosaccharide monoglyceride chain to come together and form a lipid droplet. So these we can see this is the micella, and these are all the fatty acid mono glyceride chain that come together with the help of bile. And when all these fatty acid means a chain come together, so what happened? The all the fatty acid chain would clump together, and um, they are having fatty acid core, and the exterior would be the water soluble means water soluble environment. However, in the interior they will be all hydrophobic, because in the interior all the monosaccharide chain monoglyceride chain means clump together, and they form the spherical shaped droplet. Actually, micella is a droplet of the monoglyceride chain of the fatty acid, and they form by the with the help of the with the help of bile. And what does the bile do here? Bile help these fatty acid chain uh, come together and form a droplet that is having the hydrophobic core, and exterior would be the water soluble environment. So with in this form so fatty acid in this form of micella easily transport from the uh, intestinal microvilli to the cell, where to the site of the cell where they have to there where they have to. So, dear student, this was a whole about our today's lecture. Here we studied about the digestion of fats. So, in my coming lecture, we would be studying about absorption of fats, and then transportation, and then metabolism. So, before thank you very much. But before going, I would first wish to conclude about the today's lecture. In today's lecture, we studied about the digestion of fat, and in that digestion, we study about the digestion of triglyceride, phospholipid, and cholesterol. And this is, I again misconclude for you this picture again here. So what happened when fat come in our mouth, come into contact with our mouth, whatever food we take from outside, it containing fats in the form of triglyceride, phospholipid and cholesterol in our mouth. What happened, we are having here um, lingual lipases that convert triglyceride into that means remove these diglyceride, this triglyceride that remove fat in the form of triglyceride, or in the form of triglyceride phospholipid from the watery part of the saliva. And what happened, that phospholipid help to perform this task, means to remove the fat from the water, and then watery content. And then what happened, then this triglyceride reaches in our stomach, and in stomach gastric lipases work on that triglyceride and convert into diglyceride. And this this diglyceride uh, reaches to the intestine, and in intestine pancreatic lipases work on the diglyceride and convert them into monoglyceride. So in convert them into monoglyceride, and these monoglyceride then transported in the form of the mesalie uh, in into the towards the site where they have to absorb. So this was about the whole today's lecture. Now, question is just how the monoglyceride can be transport from the intestinal lumen to the into the microvilli cell, and then how can they can be absorbed into the lymphatic vessel. So here we can see from this picture the fatty acid and the monoglyceride that are present in the intestinal lumen will add up with the emulsifier, and they form the mesola, and then these mesola will enter into the microvilli cell. And here in the microvilli cell with this mesola, the other triglyceride, lipoprotein and cholesterol component or derivative would add up with this mesola, and they form chylomicron. That is a major molecule, lipid molecule that are having lipoprotein core. So these these chylomicron then ejected by the process of exocytosis from this cell, and then enter into the lymphatic vessel, and from where they transported into the blood via the jugular vein of the neck. So this was the whole about the absorption, transportation and digestion of lipid. So here we can see, for your more information, I have put a diagram here of the chylomicron. What are the chylomicron? The chylomicron are small fat globule composed of protein and fat. What type of fat they are mainly having? They are having triglyceride, cholesterol, triglyceride molecule, cholesterol molecules and other lipids. So the one of the most important disease that is occur due to the poorly absorbed cholesterol is the steatorrhea. So this steatorrhea can result from the disease that affect absorption, such as Crohn disease and cystic fibrosis. So this steatorrhea is one of the disease of lipid malabsorption. When the lipid or the cholesterol would not absorb properly, so a person is having condition that we say it steatorrhea, and this steatorrhea would further lead other disease, and cystic fibrosis is one from, of them. So this was whole about the digestion, absorption, and transportation of lipid. So these are some references. So thank you very much, students. Kindly see my other video for metabolism of lipids. Allah Hafiz.